campus there. And being in food science, I'm a little bit of an oddball in my department in that I'm the only person in that department who doesn't work with anything that ever ends up in somebody's mouth. Instead, I work with food waste. What do we do with that waste? How do we handle all the things that people don't eat? And it's my pleasure to be able to talk to you today about some of the work that we're doing in that area for sustainable management of food processing residues, particularly in the area of tomato processing. When we talk about sustainable management of any food waste, it's not just about diverting these materials from landfills and environmental stewardship. It's really about trying to take these materials and add value to them so that they're not just a waste stream for a food processor, for instance, they're more of a co-product stream. So taking something with no value and squeezing every ounce of value that we can out of it, whether it be by turning these residues into bioenergy or otherwise converting them into other high-value co-products. For tomato processing in particular, the primary waste stream from that industry is something called tomato pumice. This is the byproduct of tomato paste production. And what this is all the skins and seeds primarily that don't end up in the paste. They're separated from the juice before it's condensed into paste. And this is a major waste stream in California. California, as you might know, is responsible for 90% of the state's processing tomato production. And with that comes a tremendous amount of tomato pumice. This state generates about 60,000 dry tons of this material every season. And while some of it may be sold for feed, other times it's composted or even taken to landfills where it's an environmental cost and monetary cost for the food processor. So we'd like to do something with this tomato pumice, see if we can find a high-value application for it. When we talk about tomato pumice, at the molecular level, this is primarily comprised of a material called lignocellulose. And lignocellulose is made of several primary components. What I've shown here in stylized form is cellulose microfibrils. These are the primary structural element of the pumice. What you see here are polymers of glucose that are wound into microfibrils. This confers strength to the plant cell wall, and on top of that, these microfibrils are cross-linked by other sugar polymers called hemicellulose. Oftentimes this is xylose polymer, but it can also be polymers of arabinose and mannose. If you think of the cellulose microfibrils as kind of the steel I-beams that support the structure of this material, this blue hemicellulose would be the rebar that keeps those main structural elements in place. And then in addition to these polysaccharides, there's another compound called lignin, which is a highly asymmetrical, highly branched polymer of polyphenols that acts essentially as the cement that glues this whole structure together. And this is a cellular structure that evolved specifically to resist degradation, but degradation is what we must do if we want to get at that cellulose and unlock the glucose from it, because it's that glucose that we can convert pretty much into any product that microbes are able to ferment. So that could be biofuels, it could be bioplastics, it could be surfactants, enzymes, other high-value chemicals. So if we want to get high-value co-products out of this material, we need to get at that cellulose. We need to figure out a way to effectively and efficiently deconstruct it to access the glucose that's locked within it. And this is not a challenge that's unique to just tomato processing. This is a hurdle that you encounter really with any lignocellulosic waste. And most fruit and vegetable processing wastes fall into this class of lignocellulosic wastes. The good news is that nature holds the key to deconstructing lignocellulose. You can think of a lot of environments where plant biomass is decomposed, and one of those environments would be a compost heap, not unlike the one you might have in your backyard. If you think about what's happening in compost, it's an environment where plant material, could be yard clippings or food waste, is rapidly broken down. And to do that, the microbes that live within the compost microbial community must be very adept at deconstructing lignocellulose. And not only do they do that efficiently, but they do that across a wide variety of environmental conditions. Large swings in temperature, oxygen content, moisture content. So the microbes that are able to deconstruct lignocellulose in compost are incredibly robust. And we need to learn how to do what they do if we, as waste managers, want to deconstruct this material and bioconvert it into co-products. So with that in mind, with the goal of trying to discover what microbes within a compost microbial community can effectively deconstruct tomato pumice, we set up several bioreactors in what's known as an enrichment culture system. In enrichment culture, the goal is to apply culture conditions that select for a functionality of interest. So in this case, our target functionality is the ability to degrade the pumice lignocellulose. With that goal in mind, we set up our reactors to utilize the tomato pumice as the sole carbon source, and then we inoculated it with our stable compost, and we added a carbon-free nutrient media to provide all the nutrients that those microbes would need to thrive, aside from carbon. So if any microbe in that compost wants to grow, the only way it can get carbon is by deconstructing the pumice and exhibiting the functionality that we're interested in. For our enrichment culture system, we set up several culture conditions with the hope that we could isolate different microbial communities that might have optimal deconstructive activity. Different microbes prefer different environmental conditions, and so we wanted to cast the broadest net that we could. To do that, we used two temperature conditions, broadly a mesophilic condition at 35 centigrade and a thermophilic condition at 55 centigrade. And then two aeration conditions, so an actively aerated aerobic condition and an oxygen-deprived anaerobic condition. Over time, we measured several responses, the first being endoglucanase activity. Endoglucanase is a type of enzyme that degrades cellulose, so it's right in line with the functionality that we're interested in. And then we also looked at the phylogenetic composition of these communities. So if we ended up with a community that could effectively degrade lignocellulose, we wanted to identify, ideally at the species level, who those microbes were. So here's some enzyme activity data after 2 weeks of culture. As I mentioned, we assayed for endoglucanase activity. Endoglucanases, shown here in blue, are enzymes that are able to bind to the surface of those cellulose microfibrils and create nicks in the cellulose chain. And that helps make the material more accessible to other cellulose degrading enzymes. The plot on the right shows the endoglucanase activity for each of our 4 enrichment cultures. So you can see that there was significant activity in all 4 of those cultures, which means that we ended up with 4 microbial communities that could, in theory, degrade tomato pumice, and were confirmed to produce at least one of the target enzymes that we were interested in. From there, the question became, well, who are the bacteria that are in these communities, given that they have this functionality that we're interested in? Who might be responsible for that functionality? To answer that question, we used next-generation DNA sequencing, specifically 16S ribosomal RNA gene sequencing, which is one of the emerging massively parallel high-throughput sequencing technologies for characterizing microbial communities. And it really capitalizes on the fact that every bacterium, every species of bacteria, contains this one gene called the 16S ribosomal RNA gene. So it's conserved among all bacteria, but it contains little fragments within that gene that are called hypervariable regions. These are essentially spacers within the gene. They need to be there at a certain length, but it doesn't matter what the sequence of those spacers is. So these variable regions accumulate mutations over time, and these mutations are species specific. So if we look at the sequences of these variable regions within this gene, it allows us to look at essentially a genetic fingerprint that we can cross-reference to databases of all sequenced microbes and predict, at least at the genus level, what are the constituents of these microbial communities. That's exactly what we did for these communities. Here are the data from that. What you see on the left is a diagram called a non-metric multidimensional scaling plot. And this is a graphical way to communicate dissimilarity between different microbial communities. So samples that are spatially closer together on the plot represent communities that are more similar in their microbial community structure. Samples that are further apart represent greater dissimilarity between the microbial communities. And what you can see here down in the bottom left corner, that's the community structure of the starting compost. And then you can see that there are distinct, ecologically unique communities that evolve for each of our target culture conditions, whether it be thermophilic or mesophilic or aerobic or anaerobic. The plot on the right shows the phylum makeup of these microbial communities. The leftmost column is the phylum diversity of the initial compost. You can see it's quite diverse and relatively even between most of the phyla represented. That's in contrast to our enriched communities. You can see that there's heavy enrichment for certain phyla under each of the target conditions. Broadly, for the aerobic culture, you can see a strong enrichment of proteobacteria, and that's in contrast to the anaerobic cultures where there's a stronger enrichment for the Firmicutes bacteria. So we're starting to hone in on who potentially might be responsible for degrading the tomato pumice in each of these communities. And if we look specifically at the genera that are enriched in each community, we see some genuses that are familiar, like Streptomyces and Ruminococcaceae. These are bacteria that we've found previously in the bovine gut, the termite gut, in forest floors, all environments where you would expect plant biomass to be rapidly broken down, and these would be bacteria that produce enzymes that help accelerate that degradation. But there are also some interesting genera that are enriched that you haven't seen as much research regarding. For instance, the alkali limnicola that are found in alkaline marshes. Nobody has really looked at those from the perspective of lignocellulose degradation before. So it's very helpful to start to look at what are the bacteria that flourish under our target conditions, because if they're able to grow effectively under those target conditions, it's very likely that they have the ability to access the carbon within the tomato pumice and produce enzymes that degrade tomato pumice. But we do want to eventually get down to the gene and enzyme level to figure out what are the molecular tools that these bacteria use to degrade this material and unlock the fermentable sugars. So there are new software tools that allow you to do this. This is the output of one such tool called PyCrust. This is a tool that takes the list of bacteria that you've identified through 16S sequencing, and it cross-references it to databases of all sequenced microbes. And then it can filter those genomes for either direct hits that match the bacteria in your community or closely related bacteria. It can filter those genomes for genes of interest. In this case, that would be genes that code for enzymes that are known to have lignocellulose degrading activity. So in this heat map, in the column to the right of the plot is a color coding that for each gene family indicates the substrate that it acts on. Does it degrade cellulose, hemicellulose, lignin, pectin, etc.? And then within the heat map itself, the intensity of the blue color represents the amount of genes that code for that gene family within the community. So broadly, you can see that in our aerobic community, you see a greater relative abundance of enzymes that code for lignin degradation, and that's in contrast to the anaerobic communities where you have more bacteria that contain genes for cellulose degradation and hemicellulose degradation. So this right here is starting to create interesting notions for how we could exploit these communities. Perhaps there are members within the aerobic community that are more efficient at lignin degradation, and that's different than the thermophilic community that's more efficient in cellulose and hemicellulose degradation. So we're starting to see from which organisms we might draw certain enzymes to enhance deconstruction of the tomato pumice lignocellulose. So where do we go with this type of data? Now that we've probed down to the gene level for communities that break down tomato pumice, we can start to pluck out certain genes that we know the sequence for, and we can express those enzymes in isolation. We can start to look at novel synthetic combinations of either these microbes or the enzymes that they produce to try to come up with cocktails that are very efficient in breaking down our target waste stream under whatever condition that we're interested in. Ultimately, the goal, as I said, is to deconstruct this material into sugars that we can ferment to other high-value co-products and divert these materials from landfills. So given that we want to turn tomato pumice into various products as a waste management strategy, one technology that you might often hear about is something called anaerobic digestion. It's one of the more mature waste-to-energy technologies out there. And in essence, an anaerobic digester is a tank, and what goes into the tank is your target waste. And what comes out are two things. One is your product, the biogas, and that's primarily methane as your biofuel that you can burn for heat, or you can combust it in a generator for electricity, or combine heat and power, whatever you like. And then the other stream that comes out are your digested solids. And so the digested solids are going to be less massive than the waste that went into the digesters. So you're doing a waste reduction step, getting some biogas out as a product. And we're seeing more and more that these solids that come out of the digester have inherent value of their own as potential fertilizers and soil amendments. So for an anaerobic digester, you have a tank, there's a mixer, there's probably a heat exchanger, but in terms of what the real interesting inner workings of this digester are, those really happen on the biological side at the micro scale. So what's going on within these anaerobic digesters? There's a complex network of mass transfer steps that define the anaerobic digestion pathway, and it all starts with hydrolysis. And during hydrolysis, certain bacteria within the digester are going to bind to organic matter, and they're going to hydrolyze it into simpler compounds. So they're expressing glucanases, proteases, lipases, and this is all turning polysaccharides into simple sugars, polypeptides into peptides, and fats, oils, and greases into lipids. Those simpler compounds are then uptaken by other microbes within the digester in a step called Osito Genesis. So during acetogenesis, all these simpler compounds are fermented into organic acids. And these organic acids include acetic acid, butyric acid, propionic acid. You also get some alcohols sometimes. And then there are several gaseous byproducts as well, including hydrogen and carbon dioxide. The next step of anaerobic digestion is acetogenesis. And during acetogenesis, all the volatile acids that aren't acetic acid get converted to acetic acid. And so the propionate, the butyrate, and the alcohols are fermented into acetate and some additional hydrogen and carbon dioxide. And then finally, the Archaea being a unique kingdom within the tree of life that exclusively houses the ability to evolve methane, takes up the acetate, the carbon dioxide, and the hydrogen, and finally converts that to our biogas, which is primarily methane and some carbon dioxide. So you can see that there's a lot of interdependencies in anaerobic digestion, and so if we ever want to change or modify this process, it's a tricky affair, because if you disrupt or inactivate any step in that pathway, the whole thing doesn't work and you don't get any methane. So when we had the goal of trying to adapt anaerobic digestion to tomato pumice and other food wastes, particularly under high solids conditions, it was a tall order, because as you start to stack unique stresses onto this digester, you increase the likelihood that you're going to inhibit one of those steps in the pathway. Particularly if you're targeting minimal water use. In this case, we were trying to get 30 to 40% solids loading, so at that point there's no free water. It essentially looks like slightly moist biomass. So you can imagine that there's mass transfer limitations, there's drying stress, there's an increased risk of inhibitor accumulation, so all things that could inhibit a digester community. So we wanted to not just use an existing digester as the starting inoculum for this project, we wanted to think, where could we find in nature methane-producing microbes that would be tolerant to high solid stress. And if you think about it, manure fits the bill. There's methane that comes off of manure, and manure, we hope, is a high solids environment, and particularly manure that's dried out in the field. So what we did is we went to the UC Davis farm, collected the driest manure that we could find, and then tried to revive that and adapt that to our target condition of thermophilic anaerobic digestion on our target waste streams. So here's some methane production data from our high solids manure that we tried to adapt to both tomato pumice and grape pumice. And grape pumice is just the skins and seeds that you have left over from wine production. And you can see on the left, the first plot is the methane production right after we added water and gave it a little bit of our pumice to chew on. Long lag period, you can see that initially perhaps it was more compatible with the grape pumice, but over cycles 2 and 3, where we continued to increase the solids loading and the pumice loading, both the tomato and grape pumice acclimated very well to the community, and we got fairly good yields of methane for high solids conditions, not quite as high as you would have for a low solids digester, but nonetheless, the point was demonstrated that we could achieve a viable, consistently performing anaerobic digestion community that could turn tomato pumice and grape pumice into methane with minimal water use. So as before, the question is, who are in these communities that are responsible for this activity? If we don't answer that question, then this whole thing is just a black box. And waste comes in and biogas comes out, and that's useful, but it creates challenges if we ever want to reproduce this or try to further improve upon our high solids anaerobic digestion community. So to get at who is in these communities, we use that same 16S ribosomal RNA gene sequencing technology that I mentioned earlier to characterize who are the bacteria and who are the archaea that persist in these enriched sludges for high solids anaerobic digestion. On the bacteria side, we saw that in general, all of our sludges were enriched for certain Clostridia. Clostridia are bacteria that are known to degrade cellulose and also ferment acetic acid, along with other cellulose degraders that fall into the actinobacteria and proteobacteria. And then on the archaea side, and these are the microbes that actually produce the methane, we found that the community was dominated by Methanocalaeus. So what these data allow us to do is to start to isolate who are the key players in these communities that we can use as a platform to further design and enhance even more functional and stable microbial communities to accelerate and advance the conversion of our target waste stream to biofuel. So I've talked a lot about how we can exploit microbial communities to help aid in sustainable waste conversion, but there's also some things that we can do directly to the biomass to help aid the conversion of this material into various co-products. So as I mentioned, most food waste is comprised of lignocellulose if it's fruit or vegetable derived, and there's a certain technology called pretreatment that tries to disrupt this highly ordered lignocellulose structure so that enzymes can more easily access that material and degrade it into fermentable sugars. And one of those emerging technologies is something called ionic liquid pretreatment. Ionic liquids are unique chemicals that are salts that are liquid at room temperature. So very different than when you think of table salt, sodium chloride, or pretty much any other salt you may be familiar with, which are all crystalline at room temperature, and even at elevated temperatures remain solid. These are salts. The ionic liquids are salts that are actually molten, they're liquid at room temperature. And so what happens if you take something like lignocellulose or plant biomass and put it in ionic liquid is that you essentially dissolve the plant biomass. And that happens because the ionic liquid has such strong ionic strength that it starts to compete with all the hydrogen bonds that keep the lignocellulose in this highly ordered crystalline structure. So if you put biomass in lignocellulose, the hydrogen bonds that cross-link the hemicellulose to the cellulose microfibrils are disrupted, along with the bonds that keep the lignin in place. And so you strip away all the hemicellulose and lignin, and then also, because the microfibrils themselves rely on hydrogen bonds to keep them in that tight structure, as the ions within the ionic liquid start to compete for those hydrogen bonds, we see a loosening of the microfibril structure. So the result of ionic liquid pretreatment is a very amorphous mass of cellulose, and you can imagine that something like this is much easier for a cellulose degrading enzyme to work its way into and access its substrate compared to that highly ordered, very recalcitrant starting structure of the lignocellulose. So what happens if you put tomato pumice in ionic liquid? This is just a series of images that give you an idea of what the transformation looks like. On the left, you have the raw pumice as it appears coming out of the processing facility. And you could put that into the ionic liquid. In this case, it's an IL called 1-ethyl 3-methyl imidazolium acetate. When you apply a little bit of heat, the third image shows that the biomass dissolves into the ionic liquid. And then the last image shows what happens when you add some water to that mixture of ionic liquid and pumice. The water prefers to interact with the IL compared to the cellulose, and so the cellulose precipitates out into the solids that you see in the end, and then we can wash everything else away to isolate the pretreated biomass.